everyone, we're going to talk about sex cells, which, although they are less recognized in pop culture than the actions associated with them, are still very important to every man, woman, child, mammal, reptile, amphibian, bird, and the occasional plant on the planet. One question a brainiac like you may be asking is, well, how do we make sex cells? Well, what a wonderfully pointed question, audience member. Sex cells replicate in a process known as meiosis, the lesser broadcasted hiding in the shadows cousin of mitosis, which is the process with which all other cells besides sex cells use to reproduce. Meiosis begins with interphase, where the DNA is copied, leaving two sets of DNA. The next phase of meiosis is prophase. DNA condenses into chromosomes and then pair up and share genetic information in a process called crossover, which is very important for genetic variation in individuals. Next, chromosome pairs line up at the equator of the cell in the metaphase stage, and the meiotic spindle fibers attach to each pair of the chromosomes. Then the pairs of chromosomes are pulled apart by the meiotic spindle, and each side of the pair goes to opposite sides of the cell in a process known as anaphase. In telophase, the chromosomes finish up moving to the end of the cell, and the cell cleaves into two separate haploid cells in a process known as cytokinesis. Haploid just means that there is only one set of alleles for each trait, instead of two sets of alleles, like in cells divided by mitosis. And so that's all there is of meiosis. Part 1, that's right, boys and girls, there's an entire second part of meiosis, known as meiosis 2. Super creative, I know. The first step in the second part of meiosis is prophase, where the daughter cells condense into chromosomes again. Then, in metaphase 2, the chromosomes in each of the daughter cells made in the last division line up at the equator of their respective cell. The meiotic spindle fibers then stick to sister chromatids. Next, in anaphase, the spindle fibers pull the chromatids apart. This is the point where the chromosomes are now separated from each other. Finally, telophase happens again, moving the chromosomes to the end of the cell, and cytokinesis happens, dividing the cell in half. In total, meiosis produces 4 haploid cells that have the potential for creating life. For example, frogs have 26 total chromosomes, or 13 pairs. So, in interphase 1, when the DNA is copied, the main cell temporarily has 52 chromosomes. After cytokinesis, each cell has 26 chromosomes, and after cytokinesis 2, each cell has 13 chromosomes. So, when the haploid cells combine, 13 plus 13 will equal the total 26 chromosomes in a normal diploid cell.